Good morning, everyone, and welcome to the MGFA National Patient Conference. I am Samantha Masterson, and I serve as MGFA's President and CEO. I am so thrilled to be here today together with all of you. On behalf of our board of directors and our staff, we thank you for joining us and for supporting this important program. As you can see, we are virtual once again, and we just really appreciate your ongoing support as we navigate this pandemic. This conference is so powerful for so many reasons. We are able to bring key stakeholders from across the MG community together, ranging from medical professionals to volunteers and donors, to strategic partners in industry, and of course, patients and their caregivers. We come together to learn more about the MG landscape through learning from top experts in the field, as well as learning from each other through Q&A, chats, and sharing our collective experiences. Today, we are going to be learning from experts on topics such as navigating the ER and other emergencies, caregiving and navigating a crisis, the rare of the rare MG, pediatric, and seronegative. We will also be hearing from patients and learning about their journeys, so we are really excited for today's program. Please take some time to visit our sponsors in the exhibit hall. You each have a virtual briefcase that you can fill with any materials from the Resources Center that you find helpful. And you will be able to access presentations and your briefcase after the conference. You can also go to the lounge and interact with each other and visit the MGFA booth. We hope you do this. We hope during our breaks, you will be able to explore the environment, the full environment. There's a lot to see. Please note that you will need to leave and then re-enter the theater in order to view each new presentation. For those of you who joined us the last couple of years, this will be familiar to you, but for those of you who are new, you just need to leave the theater each time once a presentation ends. You can go back out to the lobby, you can go to the Resources Center. There's a lot to see and to explore, but you must re-enter the theater to see a new session. So just remember that. Okay, so before we dive into today's program, we would like to take some time to remember someone very special to the MG community who left an indelible mark on so many, and that is Nancy Law. Nancy passed away last September. She most recently served as the chair of the MGFA Board of Directors, and previous to serving as board leadership, Nancy served as MGFA's CEO from 2016 to 2020. And previous to that, she served as a board member for many years. She also was a beloved volunteer of the organization for decades. She was a friend and a mentor to so many, including myself. And I know that many of you knew Nancy or knew of her. And I want to acknowledge that her passing has been felt throughout the entire world in the MG community, and what a significant loss we have all endured. I'd like to ask that you all join me in taking a moment of silence to remember Nancy Law. Thank you for taking this time to remember Nancy. We're going to now share a video in Nancy's memory, and then our keynote speaker, Dr. Chip Howard, will begin today's program. Thank you.
So I just said to a friend, a mother is only as happy as her unhappiest child. And 65 years ago, a mother named Jane Ellsworth was very unhappy. This is perhaps the most exciting time in research in myasthenia gravis. For 6 decades, from 1955, when Mestinon was approved by the FDA, there were no targeted treatments and therapies. There were no real clinical trials happening for agents in myasthenia gravis. And the landscape has changed, and it's very exciting. I think it gives hope for patients diagnosed today, that their experience with MG may be very different from people like me who were diagnosed 25 years ago.
Ooh, someday you wish upon a star, wake up where the clouds are far behind. Be where trouble melts like lemon drops, high above the chimney top.
Hi, I'm Nancy Law. I'm here tonight at the Myasthenia Gravis Foundation's 13th International Conference on Myasthenia Gravis and Related Disorders, and I'm here today with a distinguished scientist and physician, Dr. James Howard from the University of North Carolina School of Medicine.
Dream that you dare to, oh why can't well, I see trees of Greenland, red roses too. I watch them bloom for me and you, and I think to myself.
Part of that journey then was finding the right healthcare team to help me to manage MG, and also to be sure that I was informed about the disease myself. So I went to myasthenia.org, the MGFA's website. There was information for me there, and I also learned a lot about what I needed to do to be a take-charge patient. Because when you have a rare disease, you have to find a doctor who's knowledgeable and also willing to work with you, because all treatments don't work for every patient with MG. It's a journey to find the right treatment regimen.
Faces of people passing by, I see friends shaking and singing, how do you do? They really say, I love you. I hear babies cry and I watch them grow. They'll learn much more than we'll know, and I think to myself, what a wonderful world. Someday I wish upon a star, wake up where the clouds are far behind.
Hello, everyone, and welcome to this National Patient Conference of Myasthenia Gravis Foundation of America. I'm honored to be able to give the inaugural Nancy Law Lecture, and I'm humbled to do so. I've chosen my title based on my interactions with Nancy. Hear my voice, hear our voices. These are my requisite disclosures. And here are some thoughts I've had about Nancy: a leader, an innovator, a confidant, a tender heart, and a listener, a superb listener. These are only some of the adjectives that come to mind when I think about her and when I reflect on my years of interaction with Nancy. First, when she was the CEO of MGFA, and then as a member of the board. She is one who would never back down from a challenge, yet was readily and ably one who offered her time and her effort and that of the foundation to move the bar forward as we make progress in MG therapeutics. I'll miss her smile. I'll miss her sense of humor. I'll miss the challenges she posed to me as we worked together over the years. but most importantly, I'll miss her passion for all things MG, for her patient colleagues, her research scientists, and her clinicians and the MGFA. Nancy, you've set the bar extremely high, but we'll meet your challenge as you continue to guide us. We're going to talk about the advances in therapeutics, and much of it is based on what Nancy has been driving for and the questions that she has been posing to us, her voice in the MG community. We'll speak about eculizumab and efgartigimod, the only regulatory approved drugs to treat myasthenia gravis as of October 2017, and most recently in December. The few that you see in the middle, batoclimab, inalizumab, nipocalimab, ravulizumab, Rosie as we call it, and zilucoplan are all investigational drugs currently under therapeutic study. And any other drug I mentioned, while FDA approved, we use these in an off-label fashion. So these are some quotes from some of you out here in the audience. And it's reflective of the challenges you face and that clinicians face. I'm at the emergency room, but all my tests are back fine. I have shortness of breath and a small cough. I'm scared and upset. I don't feel good. I feel so alone and defeated with this disease. My family, friends don't provide me support. No one understands what I'm talking about in terms of my symptoms. My mother-in-law said, that excuse only lasts so long. Are these familiar? I'm a person with an invisible disease. This is what it looks like. I was diagnosed at 19. I'm now 63. It was a struggle to find the right combinations. We hear these all too often, and Nancy insisted that we do better. This is the title of a recent editorial that appeared in Neurology a few months ago. People with myasthenia are getting better, and I'll show you data to that fact. But are they really doing well? And as a result of this, we have to take a large introspective look at how we're doing. And I'll show you that on one hand, we're doing great, but on the other hand, we're not doing so well. So let's talk about what I call burdens of disease, the physical impact of myasthenia, weakness, fatigue, etc., and burdens of treatment, all of the associated bad that we do when we treat you. And this sets the stage as to why we're desperately looking for newer modes of therapy, new drugs, new treatments, etc. This graph shows us what the mortality of myasthenia has done over the last 100 plus years. And back in the early 1900s, it was almost uniformly fatal. And we made huge progress when we had assisted ventilation and when antibiotics came into play, because much of the demise was related to pneumonia, other sorts of infections, and respiratory failure. And one sees that by the mid-30s, early 40s, mortality was down under 50%. Huge strides in this disease. And over time to the 70s, we had the advent of corticosteroids, changes in ventilation, some other therapeutics, and plasma exchange. And with that, we got even better. And then as we've added new immune suppressant drugs to our toolbox, if you will, we've chipped away, and now we're down into single digits, 2, 3, 4%. Despite that, on the right-hand side, I show you individuals with unmet needs. Some of these individuals don't respond well to therapy. but I believe the number is even greater. Maybe 65% of patients have unmet needs, and I'll show you why in a minute. David Grove, who was a very active member of this organization when he was alive, is an internist from Maimonides Medical Center in New York City, cared for a large number of myasthenic patients, and this is his own experience. And what he was able to show in orange, that we clearly made progress in those who were improved and got better with our therapies. And in the teal color, those who passed away and died got less and less, and as I've just showed you. But let's look at the middle. In the lime green, the number of patients going into remission has not changed. Those who are improved, we see some oscillation in the curve, but overall is not changed. And those who get worse, maybe there's a little improvement, but it's really not substantial. Clearly telling us that we've helped aspects of the disease, but clearly we have a ways to go. Here's our therapeutic toolbox, and to many of you, you'll recognize these names. Symptomatic therapy with things like pyridostigmine, mestinon, the cholinesterase inhibitors. In some subsets, we use diaminopyridine. We then can use immunomodulatory drugs like intravenous and sub-Q immunoglobulin, plasma exchange, and more recently, eculizumab, though its use is very restricted, much more than we as clinicians want to use, but that's a payer issue for us. And then we have a whole host of immunosuppressive drugs, steroids, azathioprine, CellCept mycophenolate, tacrolimus and cyclosporin, methotrexate. Many of you, if not all of you, have had exposure. And then we have thymectomy. Rituximab sits in the middle because it straddles two classes of drugs, and we'll touch about these in a little bit. But we've got some eye-opening information, and this is a data cut through the Foundation's MG registry. And thank you for those who have participated. I urge all of you to participate in this registry. It's how we gain insight into the disease. And when we look at those in terms of their activities of daily living score, MG-ADL score, and where lower number is better than higher number, one can see that 50% still have scores over 6, which is fairly substantial impact of the disease. And then quality of life instruments, the panel on the right, one sees this broad range of responses. Some with very low scores are doing quite well, as you can see, but there's still others who have huge impacts on their quality of life, which is not acceptable to them, to their families, even to their treating physicians. When we ask the question, what are you dissatisfied with? One of the big ones on the lower left are side effects. And if we take all comers, it's about a third of patients are dissatisfied because of the adverse events one gets because of the treatments we choose. And if you're on corticosteroids, that number goes to 80%. People do not like it at all. What else are we dissatisfied with? Most of the drugs that I choose, we as clinicians choose, take some 4 to 6 months to even start working. One drug, azathioprine, may take up to a year. And then when we see truly meaningful response, on average 12, 18 months, azathioprine may be 3 years down the road. Clearly a long time, and what I call we're robbing the patient of quality of life because of this. Up to 41% don't like the effects in terms of how well it makes them, a lack of efficacy, lack of benefit. And then there's about a third who dislike or dissatisfied with the logistics of the therapy. having to go to an infusion center, to a plasma exchange center, having someone come into the home, and some of my patients describe it as invading my home by a nursing team to administer a drug. And so they're dissatisfied with that. And these are all reasonable concerns that patients have. Many of you have experienced side effects, and they're based on what drug we choose, and the severity is related to the drug that we choose. And some can be mild and annoying, like queasiness of the stomach with Mestinon, and others can be quite dangerous with life-threatening infections, or problems of our bone marrow, or our kidney, or our liver. And that's why many of you have to have blood work or some other testing done on a regular basis so that we as clinicians can monitor for this and try and minimize any risk that one has. Many of these are short-lived, but there are others that are very long. If one develops diabetes or high blood pressure or osteoporosis because of the drugs that we use, or kidney failure, or certain infections within the brain, etc. Here's some other quotes that I think are very telling. Just because someone carries it well doesn't mean it's heavy. How many times have you experienced that? People with NEACHR antibody positive MG don't fake being sick. And how many have been told, you look too well, you can't be sick? And then when we look at the impact of disease and treatment burdens on employment, to me this was a real eye-opener. Denmark, 47% face long-term sickness absence. In Thailand, 58% become employed. In Australia, 59% are unemployed. In Japan, a majority lose work hours or are reassigned to a job of lesser quality because of their disease. Significant treatment and disease impact on quality of life. So what does the burden of MG really mean? It means it can make socializing difficult, feelings of anxiety, isolation increase, long-term treatment risks. Patients may refuse therapy or not be adherent to avoid the changes in physical appearance, weight gain, bloating, acne, etc. Many of us as treating physicians have seen that. And then myasthenic crises can occur in a significant number of individuals, which can be life-threatening to the patient. So we as clinicians, based on challenges for people like Nancy, have had to step back and say, how do we do better? And one is to understand why we have MG. And there are several mechanisms by which nerve and muscle fail to communicate properly with each other, thus resulting in muscle weakness. One is that we have antibodies that bind to the receptor, the docking site for the chemical released by the nerve, that don't allow the chemical to get to its proper docking site and thereby inhibit and do not allow that muscle fiber to contract. And you can look at muscle as hypothetically as, say, having 10 individual muscle fibers, and if 5 of them don't contract, you've got 50% weakness. And so we need everybody in the game, so to speak, to maintain full strength. So antibody blocking plays a role. In addition, these antibodies link up with each other. We call it cross-linking. And it does an acceleration of a normal turnover process. These receptor complexes become internalized, degraded, refreshed, and new ones come up to the surface of the muscle membrane somewhere between every 7 to 10 days. Depends upon the muscle, depends upon the species. Animals are different than humans, and certain animals are different from others. But the net result is that the number of receptors on the surface looking to be able to dock the chemical transmitter become less. And with less receptors available, the probability of not getting that muscle fiber to contract and contribute to the overall strength of the patient becomes less. So a class, and we're going to start with the newest and go backwards a bit, is a class of drugs called neonatal FC receptor inhibitors, FCRN. So what are they? All women know about them already, though they probably didn't recognize the name. And they were first identified in 1956 by Professor Francis Bramble, and it's called the Bramble receptor. And it's what allows mom's immune system to be transferred to the fetus in around the last 2 months of pregnancy. And so when infants are born, they're immature and have not made their own immune system. But nature has created this process by which we've been able to transfer maternal immunoglobulin, good antibodies, and sometimes bad, from mom to the fetus and offer protection for the first several months until the child builds their own immune system. So this receptor was known for years, but it was not until the 1990s that Professor Sally Ward, who then was at Texas A&M, she's now in Southampton, England, identified this FC receptor not simply as a mechanism to transfer from mom to fetus, but as a global regulator of IgG levels, antibody levels, and allows the IgG and the antibody to have a very extended half-life, much longer than many other of the proteins that circulate in our system. The next step came when two fellows over in Belgium, Hans de Hard, Peter Ulrich in the lower right, developed the first inhibitor. And it was an IgG1 FC fragment, as we call it, and it was engineered to bind very tightly to FCRN and inhibit its action. So how does this all work? This is a busy slide. Don't panic. Let's look at the graphic on the right-hand slide, but the one to the left. And this is a cell. And you can see these little antibodies floating around in there. And you see them being taken up into the cell and then transported back to the outside of the cell again, put back into the circulation, if you will. And this is how IgG and antibodies extend their half-life from a matter of 1 to 2 weeks to over 4 weeks in time. So they bind to these little yellow ovals. Those are the FCRN receptors. And that FCRN transmits or transfers the immunoglobulin back into circulation. Now look at the right-hand graphic. If we can bind and block that FCRN inhibitor or that FCRN receptor tightly, then much of these IgGs and antibodies can't get transported back into the circulation and then get shunted to that little cell organelle in the lower left with all the little black dots. That's a lysosome where the antibody gets destroyed, where the IgG gets destroyed. So by blocking FCRN, we increase the probability that that molecule cannot get back into circulation and gets taken to the waste paper basket, so to speak. So by blocking FCRN, this data just shows that very early they were able to show that it worked. It worked for all IgG classes, of which there are 4, and this becomes important because AChR positive MG is predominantly IgG1 and IgG3. Musk myasthenia is predominantly IgG4. And this worked in all of them, depended upon the dose, but it really worked in every subclass. After an initial pilot study, what we call a phase 2 study, a phase 3 study was designed to see just how good this was. So some of you may have well participated in the ADAPT trial. 167 patients with mild, moderate, severe generalized myasthenia gravis who had a particular ADL score of at least 5 were allowed to enter. Now, this study was very unique in two situations. One aspect is that the FDA, in consultation with the sponsor, allowed other patients into the trial who did not have antibody to the acetylcholine receptor. This was the first ever trial that included non-AChR antibody positive patients. So patients with MuSK, with LRP4, or no identifiable antibody were allowed into this trial. They were only 20% of the population, and the FDA said we will do all of our analysis based on the other 80%, the AChR positive, but we're going to let you test out this other hypothesis to see if it would work. So patients were randomized 1-to-1 and then given an infusion once a week for 4 weeks, and then they were followed. And then we had to score the patient, and we used the MG-ADL score, which is a requirement of the FDA, but we strengthened the hypothesis. We simply didn't look at change from start to finish. We said they had to improve by at least 2 points, and they had to maintain that improvement for 4 consecutive weeks. If it was only 3 weeks, we didn't call them a responder. 2 weeks, same thing. 4 consecutive weeks. And then we looked at how many did and how many didn't. And then we had rules by which you could be retreated. And in those individuals, either you didn't respond for at least 8 weeks at all, or if you did respond, you came back to within 2 points of your pre-treatment ADL score, and then we could retreat you. And this middle graphic shows the retreatment. I'm not going to go into detail into this slide, but folks had a disease that was of significant duration, around 10 years. They had ADL scores that were severely abnormal, around 9. Their quantified MG score, QMG score, was in around 15 to 16, very substantial disease. Mostly mild and moderate disease, a few severe disease. These are tricky because they tend to be brittle and can't get through the trial. And then we had looked at what sort of drugs they were on. And this is what we found. And I want you to look at the 2 big deep graphs. The blue is the antibody level. The green is the immunoglobulin level. And you see very quickly within 1 dose, they're changing, they're getting better. And they get to their best about a week after their last infusion, and then slowly come back to the baseline. And the top 2 lines are the placebo arm of the study. And if I overlaid the ADL score here, it would be similar, but not to the same degree, which is what we expect. What we found is that nearly 70% of patients achieve what we call this MG-ADL responder definition versus 30%. In myasthenia, we have very large placebo responses, and there are a number of reasons for it. Primarily, the disease fluctuates moment to moment, and so capturing somebody at a very precise time point in their disease activity is very different because it's changing. So we have to deal with these very large placebo responses, and we'll touch upon that in a bit. When we look at this quantified MG score, which is what we call a secondary analysis point, you see 63 versus 14%. So both very supportive of active drug. 85% of the patients who did respond did so within 2 weeks. Not 4 months, not 6 months, not 12 months, 2 weeks, which is very gratifying. We now have something that works quickly. Before, we did not. Prednisone came the closest, on average somewhere between 3 to 6 weeks before we saw treatment effect. And then we said, is this repeatable? And so the left side of the slide and the left portion of that is what I just showed you, cycle 1. If we look at cycle 2, for those individuals who met the criteria, very similar numbers, 70% versus 26% got better. There were a few who didn't get better in the first cycle who did in the second cycle, which tells us that we simply can't abandon this drug if we don't see the kind of response we want after the first 4 infusions. And then I've simply graphed this on the right-hand side of the slide, cycle 1 at the top, cycle 2 at the bottom. The dark line is the active ADL score. The dotted line is the placebo response. And one sees that they're very similar, perhaps a little more so in the second cycle. Is this additive? So as more we give, the better we get? I have no idea. We don't have any data to that effect. This is quite similar to plasma exchange. And so here's a population of patients that Jeff Guptill and I studied in looking at their responses with plasma exchange, which is the gray box inside the slide. And one sees very similar reductions in the ADL score and the QMG score. Over time, because of repeated therapies and other therapy, steroids, mycophenolate, etc., the score gets better. But within the plasma exchange, we see a very similar response, much like this is a chemical plasma exchange, if you will. This is just to show that if I make the rules even tighter, ADL at the top left, QMG at the bottom left, the tighter or the more restrictive I make in terms of the point changes. 5-point change, 8-point change, 10-point change. The dark blue is the active drug, the pale gray blue is the placebo, and one sees that more and more patients are responsive to drug and the placebo starts to melt away. And on the right side of the slide, I show you a new metric we're using called minimal symptom expression, and others may speak about this later in the conference, in which it's a ADL score, but only of 0 or 1. And you can see clearly that this drug, 40%, hit that MSE score, if you will, versus 11% of the placebo, telling us that this is very effective. Now, the other unique aspect of the trial was, having listened to focus groups, to patients and others, was we want independence. I don't want to be treated all the time. I don't want to be treated when I need to be treated. So we said, all right, then we put in this ADL retreatment criteria where they had to come back to within 2 points, etc. And this allowed us to look at how durable the drug was. And one sees that more than 50% had responses that lasted greater than 8 weeks. And more than a third lasted 12 weeks or more. And in fact, half that group went 26 weeks and didn't require retreatment. Yes, there were some that required treatment within 4 to 6 weeks, about 10, 11%. But one sees that there's a difference, telling us that we may have the capability to individualize, target and individualize treatment, precision medicine, if you will. Safety was quite nominal. They're evenly matched in terms of the adverse events that we call them, headache being most common, in most instances mild, often not even treated, runny nose, nasopharyngitis in about 10-18%. That infusions were well tolerated. In fact, more infusion-related events in the placebo arm rather than the active drug arm. Yes, there were slightly more overall infections in those who got the drug. These were mild, maybe a urinary tract infection, maybe a bronchitis. I think the numbers are too small to be very dogmatic about this, but I would say in general, the safety profile is better than any other drug in our toolbox that we're currently using, with the exception of complement inhibitors. There are other companies in play, and this is another one called rozanoliximab. This is their phase 2 data. They've just finished their phase 3 data. They don't report any numbers, but they tell us the study was positive. So there's a second FcRn drug that's available. There are a couple of other companies involved here. They're still in phase 2 trials, and one has an ongoing phase 3 trial as well. So there's a lot of interest in this methodology of treatment. Some are administered intravenously, some are administered subcutaneously, some are full-size antibodies, the majority of them, and the one I just spoke about with you is a fragment. Now, it's great that we can target the antibody and take it out, but it doesn't explain what you see here in this graph. On the left is a normal nerve-muscle junction. The bottom part, the left lower corner, is the muscle itself, what we call the post-junctional folds. The dark band are where their antibodies are or would bind. But on the right, you don't see those. They're gone. Why? Blocking antibody, dealing with antibody, cannot explain this. And this is information that was first seen in the 70s by Dr. Andrew Engel at the Mayo Clinic with his elegant work, and has been subsequently elucidated and then resulted in the FDA approval of a complement inhibitor, eculizumab Soliris, to treat MG. We now believe that when antibody binds to the receptor, the antibody is the upside down Y, the receptor are these two pairs of bluish colored logs sticking out of the muscle membrane. It activates part of our immune system called complement. And that goes through a cascade of events that leads to the formation of what we call the membrane attack complex, or terminal complement complex, which is that ring structure you see in the lower right. That ring structure literally drills a hole through the tissue, whether it be red cells in paroxysmal nocturnal hemoglobinuria or the muscle cell in myasthenia. The contents leak out and the muscle fiber dies. So the thought was, can we target part of the complement system and prevent this from happening? So work that started in 2007, that ultimately finished in 2017, that's how long it took to do it, developed a complement inhibitor targeting the complement protein 5 that would then prevent the formation of this membrane attack complex. And in the graphic on the right, one sees the normal post-junctional folds, and then they become simplified, and you see the green-like structure. And so by targeting complement, we can prevent its formation and preserve these folds. So let's talk about complement inhibition. You've heard about this before. And the primary study was called REGAIN, and some of you were probably participants in this. And then there was an open label extension. Red is placebo, blue is the active drug, and one sees that very quickly, within a dose, we're seeing that blue line get lower, which is the ADL score, the primary endpoint, and then it is maintained throughout the course, reaching its bottom somewhere around 12 to 16 weeks. The placebo, like I've told you before, they got better too. But when we roll over into the open label, and that's marked by the vertical dashed line, we see that these people also have a very rapid improvement in their score that then parallels those who are on active drug from the start. We have a new complement inhibitor, ravulizumab. That study was completed in July. Their release of what we call TLR, top-line results, demonstrated its efficacy. It's a cousin to Soliris that's been modified, so it only has to be given every 8 weeks rather than 2 weeks. And this will go to FDA approval probably sometime towards the end of the year. There is a third complement inhibitor, and this is called zilucoplan. This is their phase 2 data, though their phase 3 data was announced last Friday as also a very positive trial, and it too will go to the FDA towards the end of the year for approval. But one sees a very similar response. Now the timeline is spread out. Rather than 26 weeks, we're looking at 12 weeks to start, but we see a very rapid improvement here. Red is active drug, blue is placebo, and then the placebo arm also improves. But the key is rapid, sustained response. What's interesting from the Soliris trial, the eculizumab trial, and the dashed horizontal line near the bottom is the blinded portion of the study, and the placebo is in pink, and then above that is the open label. We see that increasing numbers of patients get better over time. And when we look at those who we say have minimal manifestations, that is, no functional weakness, they go from about 25% in the blinded phase to up nearly 69% in the open label extension. Are we rebuilding these neuromuscular junctions? Is there some other repair process going? We don't know yet. It's under study. But something interesting is happening, that with long-term chronic therapy, we're modulating and changing something so that more and more of these people who had failed everything, remember, Regain was only allowed to be in those patients who were refractory to treatment, they're getting better. There are other ways we can address the situation. One is called B-cell depletion. The drug you know about is rituximab. Many of you, if you have musk myasthenia, have been treated with it. Some of you with AChR-positive MG have been as well. But in AChR-positive disease, the trial did not prove to be that helpful. It's what we call a trial failure. And there are a number of reasons for that. But one of the possibilities is that we're not treating the right cell. And in the teal blue at the bottom, where it says CD20 expression, those are the range of B cells that rituximab targets. There's another drug that's currently under trial, inalizumab, that's already approved for another disease, a CD19 inhibitor, has a broader range of target. And it may be this drug will be effective in AChR-positive MG, and that's currently under study at this point. A new technology that is now being looked at is one called CAR-T. You've heard about this in the treatment of cancers. There are different forms of CAR-T. This one is designed to look at part of our genetics called RNA. It gets re-engineered to target a very specific cell marker on a lymphocyte. So we harvest the patient's lymphocytes, we send them off, they're engineered, we bring the patient back and we reinfuse them, and we see how they do. There are two companies in MG interested in this. One is dealing with MuSK, the other is with AChR myasthenia, both who are AChR positive as well as those who are seronegative. And so we wash them out following plasma exchange and IVIG, and then they're being dosed weekly 6 times with this engineered product. 3 patients with MG have been treated so far, and one sees the response in some of the outcome measures in dark blue. As reference, they're using the ADAPT trial, this FcRn placebo response, and that's shown in the red, but one sees clear separation of the lines. And so this is very exciting and very hopeful that we may have another targeted therapy. This form of CAR-T does not totally deplete lymphocytes, and so many of the potential side effects of other forms of CAR-T therapy are not seen. And theoretically, this almost could be done as an outpatient in the clinic sort of infusion ultimately. So, where progress have we made? Targeted immunosuppression, and that's going to reduce side effects. It's going to improve the rapidity of the onset of action of these drugs. We've had treatments for refractory MG with eculizumab. Understudy are different formulations, sub-Q versus IV. We have trials going in pediatrics. We've just completed a Soliris eculizumab trial in a pediatric population, and we'll be starting trials with efgartigimod and I presume with ravulizumab and zilucoplan in the near future. We need further information on the childbearing patients in pregnancy and safety. There's preliminary work with complement inhibitors that suggests they are safe during pregnancy. And we need drugs to look at for pending crisis or crisis. The fact that these two classes of drugs work so quickly, they may have a role in this as well. So, some more quotes. You should never feel like a favor when AChR positive GMG gets you down. You didn't make this disease. It was not your choice. You may fail a test, but you cannot fail yourself. Stay frosty. And listening to Nancy, I will not be silenced. I will continue to speak my voice. So yes, we hear your voices. The future is becoming brighter. We've made tremendous progress since 2007. There's so many things in the pipeline right now that will potentially revolutionize our treatments for MG. And for that, we're excited. I hope you are excited. And thank you for listening.
Thank you, Dr. Howard. That was an amazing talk. My name is Dove Levin, and I'm the National Senior Director for Program Management and Education for MGFA, and I will be here helping with the Q&As during the day, as well as my colleague Mike Antonellis, who will be on as well. We have a lot of questions, Dr. Howard. The first one is, it's so exciting to hear about all these new drugs, but how does one as a patient know when it's time to try one and what to try?
It's so exciting to hear about all these new drugs, but how does one as a patient know when it's time to try one?
So that's a great question. I think one has to have a relationship with your treating physician. They have to know your expectations and what you want from therapy. And through that kind of a dialogue, one can establish the game plan, if you will. And so it's different for different individuals. But the key is the communication. And we would spend time talking about the patients. What do you want? What do you need? What's your expectation? Somebody who's 95 years old and very sedentary is different from a 90-year-old who's out bird watching and traveling around the world, like one of my patients was doing. And so we may choose different forms of therapy. Am I planning a family and pregnancy? Therefore, certain drugs we have to take off the table and we'll have to choose something else. Regrettably, in the U.S., with our insurance and healthcare system, what are my financial resources? I often can get my drug paid for, but can I get paid or have my insurance company pay for the associated management for the blood laboratory studies, et cetera, that are necessary to monitor for potential adverse effects? And so all of this has to be put into the formula, if you will. What are my comorbidities? Do I already have diabetes or renal disease or, bone disease? And that too will color how I choose. So it's open dialogue between you and the treating physician that then allows the plan to be established.
So the next question, there were two questions that were similar, but how can you comment on how the new treatments will work for patients that are seronegative? And were they tested in the trials?
Can you comment on how the new treatments will work for patients that are seronegative?
So it's right. So the question is, how will these work in the seronegative population? Let's talk about FcRn first. And so that population, the seronegative, or I won't say seronegative, the non-AChR negative population, which included MuSK-LRP4 and seronegative, responded as well when we looked at the numbers as those who had AChR positive disease. The problem was that the placebo arm of that study had a much greater response than the placebo arm of those who are AChR positive. And because of that, the FDA was not inclined to include seronegative in the labeling. It was a disappointment to all of us, but it was related to the fact that numbers were small and this placebo response was large. It's a starting point, and I can tell you that the sponsor, the company, has not abandoned the seronegative population. And there's active discussion as to how we can best get data that will be convincing to regulatory authorities that it is as efficacious, as good as what we saw in the AChR population. It's going to take some time, but it's in the works. In terms of complement inhibitors, all of those trials have been AChR positive. And even though the FDA may approve a drug for just generalized myasthenia, regardless of antibody, like they did with Soliris, it's the payers that limit our use, particularly with eculizumab, Soliris, in treating patients. It's not the FDA. And they just mimic the clinical trial, which I personally don't feel is appropriate. We need trials of seronegative with complement inhibitors. It's not clear whether companies will move on that or whether we as investigators have to put a trial together. There have been a couple of us who have designed such a trial. The trouble with these is cost, and you cannot imagine the cost of pulling off one of these trials. It's north of 7 figures, way north of 7 figures. And that's an issue. How do we get it paid for, particularly if I'm going to do the trial and all? I can get the drug. Companies are often willing to give drug, but how do I get all the other stuff paid for? And that's the problem. So I don't have an answer for the complement world. For the FcRn, hopefully we'll have answers sometime in the future.
Thank you. So we have one last question before we move on. We have a lot of questions, but we can't get to all of them. So we'll try to do our best to get answers for those we don't get to. So this question is about a thymectomy. Normally in the course of treatment, is it the first step before taking medication, or do you take medication before you would consider a thymectomy?
So this question is about a thymectomy. Normally in the course of treatment, is it the first step before taking medication, or you take medication before you would consider a thymectomy?
So the question regarding thymectomy and the timing of thymectomy, it depends on the situation. So in general, in my practice, I want patients as strong as possible before I send them to surgery in order to minimize the perioperative risk, predominantly pneumonia, respiratory weakness in these patients. And so we'll treat first and then take them to surgery. There's very old data that says the earlier you do thymectomy, the better response you'll get. We're not sure if that's really true or not. And so I feel we should get the patient in the best shape possible and then go to surgery. Now, there are situations where they have minimal weakness. It's really not functional, but clearly have myasthenia. And in that case, one could go to thymectomy much earlier in the course of the disease. So it's really situational, but it's safety paramount, first and foremost. Can I get the patient off the operating table safely without new problem, new risk?
Thank you, Dr. Howard, and thank you for your time today. And we're going to go on to the next session. Please don't forget to sign out before you sign into the next session. Thank you.